Types of polymorphic sequences. There are different types of the polymorphic sequences which are also called as polymorphic markers. The important examples of those sequences, they are RFLP, that is restriction fragment length polymorphism. The second type is VNTR, that is abbreviation of variable number tandem repeats. Other polymorphic sequences, they can be in the form of STR, abbreviation for short tandem repeat sequences. Then SNP, that is small SNP single nucleotide polymorphism, and AFLP, that is amplified fragment length polymorphism. So these are all types of the genetic markers, and there are also types of the polymorphic sequences. Here we can see that molecular markers are of two major types. One is called as single locus markers and other is called as multi-locus markers. In case of single locus markers, they can be RFLP, they can be microsatellites or STR markers, or they can be STS. So these are the examples of single locus markers. In case of multi-locus markers, that can be DNA fingerprinting, those markers which are used for DNA fingerprinting, and amplified fragment length polymorphism, as well as random amplified polymorphic DNA. So molecular markers can be divided into two types, single locus markers and multi-locus markers. So single locus markers, they are RFLP, microsatellized or STR DNA. In case of multi-locus markers, they can be DNA fingerprinting, AFLP, and RAPD, which is also called as rapid. Applications of the molecular markers. There are two types of the applications. One is called as short range applications and second one is called as long range applications. Short range applications of the polymorphic sequences or of the polymorphic markers, they can be parentage determination, that is also called as paternity analysis, genetic distance estimations to estimate the distance between two genetic loci, determination of twin zygosity, sexing of pre-implanted embryos, and identification of disease carriers. So these are the applications of molecular markers which are polymorphic sequences or polymorphic in nature. These are some of the short range applications of these markers. While on the other hand, if we see what are the long range applications of the molecular markers, they can be gene mapping in case of humans or in case of plants or animals, and to study QTL linkage analysis of those traits which are called as QTLs. So long range application of the polymorphic markers can be gene mapping to identify the genes or to identify the mutations, as well as mapping of the QTLs. Along with this one, markers, they also assist for the selection of breeds, particularly in case of animals and plants, to differentiate between the good breeds and bad breeds. So there are two long range applications of the polymorphic sequences, the gene mapping as well as marker assisted for the selection of the breeds. Polymorphic DNA sequences which are present in the humans, in case of 0.1% is variation in case of polymorphic sequences, and all the variations which are seen between the individuals, they are due to the polymorphic sequences in the genome of the humans.